Disculpeu, ja estem preparats per fer la presentació. Glòria, Eugènia? Gràcies.
Hola, bon dia. Soc la Glòria Colom, responsable de projectes internacionals a l'Escola Agrària de Manresa. Hello, I'm Glòria Colom, responsible for international projects at Escola Agrària de Manresa. Amb aquesta jornada acaba el cicle de 7 jornades i 2 visites sobre alternatives a insubs controvertits en producció agrària ecològica. This conference ends the cycle of 7 lectures and 2 visits on alternatives to controversial inputs in organic farming. Els vídeos de les visites, pels que no hi vau poder assistir, estaran resumits i editats els propers dies. I totes les ponències i visites estaran disponibles al juliol a RuralCat en obert per a futures persones interessades o per a les que s'han perdut algunes sessions. Videos on the visits for those who were unable to attend will be summarized and edited in the coming days. All papers and visits will be available in RuralCat free access for future interested people and for those who have missed some sessions. Les xerrades han estat pregravades per tal de poder fer una bona traducció de l'anglès al català, però al final, al voltant de les 10:55, podreu fer preguntes directament als oradors, als ponents. Papers have been pre-recorded in order to make a better translation from English to Catalan, but at the end, more or less 10:55, you will be able to address questions to the speakers. També he d'advertir que el primer vídeo que veureu, de l'Ulrich Schmutz, és comú a totes les 7 sessions, per tant, alguns de vosaltres ja l'haureu vist. First video of Ulrich Schmutz is common for all the seance, and so some of you will already seen it. També us he d'advertir que aquestes sessions són enregistrades. I must notice that this session will be recorded. Thank you. Gràcies.
Hello and welcome to this introduction of our international course in contentious inputs in organic agriculture. It's held at the Escola Agrària de Manresa in Catalonia, and I will give you the introduction. My name is Ulrich Schmutz, and I'm the coordinator of the Organic Plus project. I'm also a professor at Coventry University. We have a center, it's called Ryton Organic Gardens, and it's a center for agroecology, water, and resilience. So you find my details there. And I will give you a little bit of an overview of this course, so then you have a good way into it in the following sessions. So welcome again. So we are a team of 2 large EU-funded projects called RELAX and Organic Plus. I'm the leader of one of them. Lucius Tamm is the leader of the second one. And together we had 8 million euros funding of 4 years. And we are now in our final year, and we have a lot of results to share with you, but we haven't solved all the issues. So I want to give you a little bit of a issue where we are now. Organic farming has phased out contentious input a long time ago. It's something which has been done, and it's part of the DNA of organic movement. So it started with phasing out chemical fossil fuel fertilizers. We don't use them, and that's part of the organic regulation. We have phased out synthetic fossil fuel-based pesticides. There's also no GMOs, and there's also quite strict regulation on animal cruelty, or on a positive word, how to care about animals. So the word organic or agroecology, biological agriculture, aims to be a fairer, more ecological, more caring and healthier system innovation. These are also the principles of the organic movement. However, it is not perfect. So we need further improvements to organic. We can't just stay there. And this is in some way the kind of organic plus principle which we want to share with you, and which you also as a farmer or advisor are part of in a way to continuously improve organic. So I'm going a little bit back in a way to set the context of this course. So let's go just back before we look forward. So organic started almost 100 years now with a movement called organic biological farming and also biodynamic farming. So we go back quite a long time. There's a lot of history there. And then in 72, there was formation of an international, so both of those earlier things were a bit more in Central Europe, Switzerland, and what is now Poland. And then there was an international movement, the International Federation of Organic Agricultural Movements, that was founded in Paris. And this was a time before there was regulation on organic farming. And this came in Europe and in the EU in 1991. And ever since, regulation has been done. So our work, in a way, is to look where we are now. So are we in this year, 2022? We have just had an updated regulation on organic farming, and obviously have to say that there is also a big war, obviously, now going on in the center of Europe, which is also an issue of some of the contentious inputs we say later, especially oil and minerals. But we'll move on with that and want to look more forward. So what was the project about? So looking forward is going to 2030. So we will have an official target from the Green Deal of the European Commission that we will have we aim for 25% organic land use in the EU. This is quite a big aim. Some countries like Germany, Austria, some smaller ones, they aim even further for 30%, and some of them have already achieved that. Liechtenstein, I think, is the highest, over 40%. So this is the big setup where we are in. And going now into the course, as I mentioned, we have those two projects. You can see their title. They both have a website where you can study and Google that in more detail. And both projects were kind of organized a little bit. We knew each other already that we were applying. It is still competitive, and other projects were not funded, but we try to be very synergistic always, so that we can kind of, in combination, can solve these issues of contentious input. So your whole course now will then tackle the contentious inputs we see now in organic farming and how we can overcome them in detail. So we have to go into the detail of copper. We have to go into the detail of mineral oils. We will present you experiments, not everything we have done in this course, but just selected information and results on that. And then we will go in the following days, you will hear more about vegan fertilizers, you will hear more about peat and the replacement of peat, you will hear more about plastic use. All of these are identified contentious inputs. And then we have a big section also on livestock farming, because there we have also a lot of contentious inputs, synthetic vitamins, antibiotics, anthelmintics, so this is a treatment against worms, and also the animal bedding, which is often with conventional straw, which then has the pesticides in it as well. And we are concerned about the animals eating them, but also contamination. We also had a big part of research on social science. We're not covering that in this course, but we will work also on the modeling, on the phase out. So you will get information on the life cycle assessment and the social assessment, social economic assessment of the alternatives, and how we can model the phase out. So this is a little bit, and we follow this same template in all the different sections you have. And then you obviously have also 2 days of on-farm visits, because all this information needs to be put in practical, real farming changes, because otherwise it doesn't really make any difference to the environment and has limited or no input, which is obviously impact, which is important. So based on the information and the work we are doing, looking at alternatives, we then make recommendations mainly to the European Union and also other sector bodies who may have standards, when to phase out some of these remaining identified contentious inputs. So, for example, and this is not, it's like a planning tool here. It's not the final answer, but we think the phase out has to be staggered. So some of the things we can phase out fairly quickly. For others, we need at least 10 years more time, research, alternatives need to be prepared. We don't want to ruin the organic movement by going too fast, but also we want to reach 30% or 25% in the next decade. So we need to do that in a growing market. And then there's others like antibiotics, which may take even longer to completely phase out. However, that may actually not be true, because depending on the research we do, it could actually also change. So this is another overview. So, and I also will finally end that we also already, because we are at the end of the project, we are thinking about the next steps. So I call them phase out mission proposals. So we want to have further research commissioned by the EU, and that hasn't been done yet. Also parts of it have. And they would be slightly different, because our work we're showing you was work which was more commissioned in research labs and trial sites. And this wider phase out would be even more connected to farmers. We already connected a lot to farmers in our project, but an innovation action would also include farmers as micro enterprises. So then we can do commission trials on farms, so that everyone is kind of has help in a way to try the alternatives and also create. We will hear, this is another word I haven't mentioned yet. For some of them, we also think you need a system solution, which means a redesign of the organic system, which in many cases is a little bit, what I could say, old-fashioned, because it was converted from conventional farming. So we have a system like in apple trees, citrus, which worked really well under the planting densities of conventional agriculture. Then it was converted to organic, and now it doesn't really work so well. So we need to have a system redesign, including agroforestry and mixed farming, so we get a whole new system where we don't have these problems. And this is all from me now. So I thank you for your attention up to now. So I would say, please enjoy the course, and obviously some of you may be more interested in horticulture and other more in livestock, and that's clear. But I think this project has also helped, because it has so many issues, help bring the organic movement together and remind everyone that there is this interconnection between mixed farming, livestock, and even in horticulture, where you can do that. And then my final word is, please also take some action. Do some experiments on your farm. Try some of these alternatives and see how how it goes. And then I hope we can create an organic plus system, a world almost, where we do not need all these fossil fuel products. Obviously, a main thing is also the climate change. Heat, for example, if we don't use it, we can protect all the peat bogs on our planet, and they have an enormous potential to mitigate climate change. So it may look like a small step for you on your farm, but it is actually quite important to come to this bioeconomy or cyclical economy, as we call it, on a larger scale. So thank you again for your interest in this, and I hope you enjoy the course. Bye.
Hi, everybody. Firstly, I'd like to introduce myself. My name is Berit Blomstrand. I'm a Norwegian veterinarian educated in Hanover in Germany in 1999. Presently, I am working at the Norwegian Center for Organic Agriculture. Previously, I have mostly been working as a veterinary surgeon with livestock, including cattle, sheep, pigs, and horses. Presently, I am a PhD candidate learning how to become a researcher, and my PhD project has been focused on assessing the antiparasitic activity of spruce and pine bark extracts against parasites in sheep. Before presenting what we have done in our project, I will have a brief introduction. Firstly, I will spend a few minutes on talking about what parasites are and why we need to act upon them. Secondly, I will say a bit about antiparasitic drugs and why we consider them as contentious inputs, meaning why we would like to avoid using them. Thirdly, I will suggest a few alternatives we have which will help us reducing the use of antiparasitic drugs. So, what are parasites? A parasite is an organism living a portion of their lives on or inside another organism, which is called the host. Ectoparasites live on the host's skin, like lice, ticks, fleas, or sandflies. Some of them suck blood, and many cause itching with or without loss of hair. Ectoparasites can transfer disease, which can cause quite severe harm, like Lyme disease or tick-borne encephalitis. Endoparasites live inside the host, for instance, in the intestinal canal, in the blood vessels, or in the brain, to mention a few. In the project I will present to you, we use the unicellular parasite Eimeria, which is known to cause diarrhea and stunted growth in young lambs. So, do parasites cause harm to the host? A parasite is dependent on the host. It lives on or in the host and takes nutrients from the host. Ectoparasites can, besides transferring disease, cause agitation and stress through itching and hair loss. Endoparasites deprive the host of nutrients and, depending on what organ the parasites infest, they can cause damage to the intestinal tissue, liver, or the nervous system, etc. Intestinal parasites are known to cause diarrhea, dehydration, and stunted growth. In worst case, they can cause the death of the host. So why do we consider antiparasitic drugs as contentious or unwanted? First, a bit about antiparasitic drugs, and I will talk about drugs against intestinal parasites in mammals only. Anthelmintics are drugs used against intestinal nematodes, or worms. There are approximately 6 groups of drugs registered against intestinal nematodes in the mammalian livestock in the European Union. In EU, 2 different drugs are registered for preventing coccidiosis in sheep, cattle, and pigs. And there are 2 drugs registered for treatment against cryptosporidiosis in cattle. So why do we consider these drugs for contentious? Firstly, the parasites develop resistance against the antiparasitic drugs. We see resistant parasites against all groups of anthelmintics and against coccidiostats, and we see it all over the world, and it is increasing. The more we use the drugs, the more parasites develop resistance. Therefore, we should avoid using the drugs where we can and save it for when we really need it. Secondly, some antiparasitic drugs are toxic to the environment, to insects or to aquatic organisms. Hence, we should avoid contaminating the environment with potential toxic substances. We want to avoid using chemicals and rather strengthen the animal's health and immunity. There are several useful ways of decreasing the use of drugs when we work to reduce the parasitic burden of livestock. The most important thing to do is to find out whether parasites are a problem or not. In Norway, the general approach is to treat against parasites without really knowing if the animals have a parasite problem. To get an overview over parasitism on the farm, it is important to take fecal samples and to get them examined at a veterinary laboratory. I would recommend you to ask your veterinarian or advisor regarding when to sample and how often. If parasitism is a problem, a very efficient way to decrease the amount of infectious parasites in the environment is to decrease the stocking rate. This implies a reduction in the amount of animals per available housing or grazing area. This is often not an option, and other measures may be better alternatives. Another efficient way to reduce the parasitic burden is to do simple things like keeping a clean housing environment for the animals. It is particularly important to clean in and around the feeding areas, indoors as well as outdoors. Increasing the time between grazing periods is an efficient way of decreasing the infection pressure on the pasture. This can be done by not using the pasture for a certain time. With time, the viability of the parasites on the pasture will decline and the infection pressure will decrease. In Norway, we advise not to use the pasture for grazing in year 2 if the pasture was used for grazing after midsummer in year 1. Advices like this are very dependent on the climate, and I would recommend you to seek advice with your local veterinarian or advisor. One way of exploiting a limited grazing area without exposing the animals to high levels of parasites is to apply co-grazing or alternate grazing with different species. When 2 species are co-grazing, for instance, sheep and horses, the horses will consume some of the sheep parasites and opposite. Sheep will remove horse parasites by grazing. Sheep parasites will not cause harm in horses, and likewise, horse parasites do not cause harm in sheep. This will reduce the infection pressure for both sheep and horses. Another way of reducing the infection pressure is having the sheep graze for a period, say 3 weeks, and then move the sheep to a cleaner pasture. The old pasture can be used for horses or cattle. This will reduce the time sheep spend in the same area and will prolong the time between sheep grazing periods, hence reducing the infection pressure. If there are only 1 livestock species on the farm, maybe there are neighboring farms where you can suggest to swap pastures and have different livestock species graze at different time periods. There is 1 vaccine available in the European Union for use against gastrointestinal nematodes in livestock, and this is against the barber's pole Haemonchus contortus. Currently, there is research going on to develop further vaccines, against important gastrointestinal nematodes in ruminants, but these are not yet available. Breeding to achieve more resistant host animals is an available method to combat parasitic infections. Animals with higher resistance towards parasites may have less need for an antiparasitic treatment. Hence, breeding for a more resistant host animal can prevent the parasite developing resistance against antiparasitic drugs. Having more resistant host animals will reduce the time and money spent on treating the animals with antiparasitic drugs. Maybe your breeding organization has a resistance against parasites as a breeding goal of their sires. This is an important moment of interest when deciding which mother animals to use in your future animal herd. Plants represent the oldest source of pharmacotherapy used by mankind. Due to the rapid development of parasite resistance against antiparasitic drugs, it has become increasingly important to conduct research on such plants. There are a multitude of plants that show biological activity against various parasites, and we have tested spruce and pine bark extracts for antiparasitic properties in ruminants and mice. And this is where I will tell you about the research we have done when assessing pine and spruce bark extracts against parasites in ruminants. Norway has a large forestry industry where trees are used for producing timber as well as in the paper industry. In 2019, the sawmills and the wood processing industry utilized a total of 7.21 million cubic meters of softwood logs, and at the same time producing approximately 721,000 cubic meters of wet bark. Today, the bark is mainly combusted for energy production, and a smaller proportion of approximately 20% is used for gardening purposes as mulching or soil improvements. There is an increasing interest in utilizing the bark for higher value applications. Bark Cure was a project we started in 2017, where we assessed the antiparasitic activity of 18 different bark extracts from spruce and pine against 7 different parasites. We implemented experiments using 6 different laboratory methods and 2 trials where we assessed the antiparasitic effect of bark extracts in animals, in mice and lambs. In the trials that we performed in the laboratory, we found that the pine bark extract reduced the proliferation and growth of Cryptosporidium parvum in cell cultures. Furthermore, we saw that several of the extracts tested reduced the viability of the intestinal worms of sheep and calves up to 100%. We also found that the bark extracts had a higher efficacy against the younger nematode stages compared to the older. This means that the efficacy was higher on nematode eggs compared to the adults. Although there's a clear antiparasitic effect on the bark extracts in the laboratory trials, we cannot automatically conclude that these plant extracts will have a good antiparasitic effect in the animals. Hence, we decided to test the bark extract with the highest effect in the laboratory in animal trials. In the Organic Loss project, we ran a trial on newly born lambs where we assessed the anticoccidial efficacy of spruce bark extract. The project was called Spruce Bark Extract as an agent against coccidiosis in pre-weaned lambs and was conducted in Norway during the spring and early summer of 2019. In this project, we hypothesized that the bark extract would reduce the fecal excretion of parasites and reduce the rate and severity of diarrhea. The bark was collected from a Norwegian sawmill in March 2019, and the bark extract was produced by Sintef. We had 3 treatment groups with 8 animals in each group. 2 groups were infected with Eimeria, which is the parasite causing coccidiosis in lambs. The third group was kept as an uninfected control group. One of the two infected groups were treated daily with bark extract orally for 11 days. The animals were kept in a parasite-free environment indoors. We took fecal samples regularly, and the consistency of the feces was assessed to evaluate the degree of diarrhea in the lambs. Additionally, the lambs were weighed regularly, and the consumption of milk was recorded daily. All lambs were slaughtered at day 22 of the experiment. Our main finding was that the lambs treated with bark extract had a reduced number of parasites in the feces compared to the untreated control group. We also saw that the difference in parasitic excretion in the feces increased with time. Furthermore, we found that the treated lambs had softer feces during the treatment period, but had a more formed feces after discontinuation of the treatment compared to the untreated group. From the figures, you can see that for the infected treated group, more lambs have a lower fecal consistency score in the period after treatment was ended, compared to the untreated group, where we can see that most lambs have a high fecal consistency score, which means a softer feces and a higher rate of diarrhea. A high fecal consistency score means softer stool or diarrhea. The non-infected, untreated control group had formed feces throughout the whole experiment. We experienced detrimental effects on the treatment. During the treatment, the lambs reduced their milk intake, and consequently, the daily weight gain decreased. After discontinuing the treatment, the lamb's appetite increased to the level of the uninfected group. As seen in the table, the growth of the group treated with bark extract was markedly lower than that of the two other groups. So our conclusion after the experiment is that bark extract from Norway spruce may be useful against coccidiosis in young lambs by reducing the amount of parasites in the feces and also reducing the rate of diarrhea. We experienced negative side effects like reduced milk intake and reduced growth. We need to work on the trade-off, meaning we need to test the bark extract in a lower dose to see if we can avoid the negative side effects and still get the lower fecal parasite excretion and a lower rate of diarrhea. We also wish to conduct another experiment where we prolong the duration after the end of the treatment. This is to assess the long-term effect of the bark extract with regards to growth. We would like to know if the reduced parasitic burden will lead to a higher body weight at slaughter with 5 months of age, which is the common slaughter age in Norway. We also need to improve the administration method. In this trial, we treated the lambs orally for 11 days. This is not viable for a farmer, and we want to assess other methods like pasta or capsules, preferably as a single treatment. That was what I had for today. I hope you found it interesting, and if you have questions, please ask now, and don't hesitate to send me an email. Thank you very much for your attention.
Good afternoon. My name is Stig Milan Thamsborg, and I'm going to tell you about alternatives to the use of antiparasitic drugs in organic pig production. And my background has been, I worked with organic farming for many years, I think since 1995, and we have focused on parasitic diseases in general, and a lot of it has been on pigs. Recently, the last 15 years, I've been working mainly on pigs, but before that, we worked on ruminants as well. And we have looked into, for about 20 years, we've looked into alternatives that you can use instead of using sort of commercial antiparasitic drugs. What else can you do and to reach a sort of balance? The outline is very briefly about organic pig production, very briefly about parasite challenges, and then I go into the topic of today, which is alternative approaches, and they will address management, pastures, houses, and manure, some dietary interventions, also something about nematode phagous fungi, that's nematode destroying fungi, and a bit about breeding. And I'll end up with some perspectives. Organic farming, there's a lot of, it may have been introduced in earlier, but there's a lot of characteristics that are sort of relevant for parasitic infections. First of all, the possibility to perform natural behavior is a hallmark of organic farming, loose herding, outdoor access for pigs and poultry. Also, health management is supposed to be disease prevention by measures of selecting the right breeds, have a balanced, high quality diet, and also a favorable environment. And that is, for example, regarding stocking density and housing and husbandry practices. Also, the preventive use of parasiticides is prohibited, but as I know, limited use is now accepted in most countries, what you call sort of preemptive use, whenever you need to do it in order to avoid disease. In practice, a maximum number of applications of anti-infective drugs per year are stipulated in some countries. And then on top of that, I think it's also very important that we have an obligation in organic farming to participate in nature conservation, sort of to run the business in sort of in consensus and in alignment with nature as much as possible. Still, we have to remember that conventional farming is also constantly changing. So the relationship between organic farming and conventional farming is also constantly changing. For example, recently, not recently, I think it's 10 years ago, conventional farming also need to have loose herding and also have to have either sprinklers or wallows for pig production. So these are some of the housing systems in organic farms or in organic pigs. The one you see to the left is where you have an indoor pen with shallow litter. The one to the right is the deep litter one, where you sort of accumulate up to 1 meter of deep litter. And then you can see this is the opening to an outdoor area, which could be either concrete with slatted floors, or it could also be, as you can see in this experimental setup here to the left, low left, you can see there are small paddocks that they can run out into. So they have to have an indoor part and an outdoor part. Farrowing is generally in Denmark on pasture. We have late weaning, more than 7 weeks. The weaners will stay on pasture for a while, or they might go into starter pens. Then we have the outdoor runs. I've already mentioned that. We have the bedding material, deep or shallow litter. Deep litter is becoming more uncommon in Denmark due to the large consumption of straw, of course. And then you have sprinklers or wallows. Organic farming is by far the smallest production in organic farming in pig industry is by far the smallest sector, I would say, in the organically produced livestock. As you can see from this, I will not go into details, but you can see I've just selected 3 countries, Greece, Austria, and Denmark, and you can clearly see that when we talk in percent of heads, it's very low, the number of pigs we have. So this is a very relatively small industry. The parasite challenges, to move on to that, is I would say that the important ones are 3 major helmets, and I'll introduce these ones because I'll get back to them for the rest of the talk today. The first one, as you can see to the left, is a large roundworm of pigs, which is called Ascrisuum. I'm sorry for this Latin name, but in many countries there's no other names but the Latin names for these parasites. But you all know that they are about 15 to 30 centimeter long worms when they are all mature, and they sit in the small intestine and they produce these awful livers because they have a migration through the liver and the lungs before they get back into the intestine. And then the livers get these so-called milk spots or white spots. Then we have the nodule, the whipworm, which are sitting in this large intestine. It's called Trichuris suis, and you can see these are long worms with a slender anterior part. This may be difficult to see on this slide, and they are attached to the mucosa, so they're hanging down. In this case, you can see, and strongly connected to the and embedded into the mucosa, into the epithelium of the intestine. And then lastly, we have the nodular worms, which are much smaller. They are 3 to 4 centimeters, and they are lying freely in the lumen, just like the ascaris. And it's called nodular worm because it forms a nodule at some stage in the intestinal wall, where they sit and until they develop and will be on the surface of the intestine. This is also in the large intestine. All of them have a life cycle where they have some, as you can see, some stages outside, either egg stages for Trichuris here, or egg stages here for ascaris, where they develop and embryonate. It takes about a month's time. And then we have the last one. This is the nodular worm, where they make larval stages. But all of these, either eggs or larvae, are ingested by a new pig and they get infected. When we look at the distribution, you can see here that the prevalences above at the top here is organic farms, and the ones at the bottom here, that's the conventional farms. And then we look at the worms, ascaris suum, that's the large roundworm, the nodular worm, and the whipworm. And you can see easily here that when you look at the number of pigs infected, it's much higher in organic herds. And these are, I know, rather old data, but this is still what we see, that you have a much higher infection level in the organic herds than you have in the conventional herds. We do this by looking at the feces, the excreta, and there you can see the different kind of eggs, very easy to recognize and easy to count. One thing that is quite interesting is, as you can see, this is a phagostomum, is typically accumulating with age. So the older the animals, the more infection you have. It's a little more complicated with for example, ascaris, where you can see most of the ones you see in the large fatteners. These are large fatteners, smaller fatteners, weaners, and piglets and sows. These are the indications. I forgot to say that. So these are the different age groups infected. Another parasite that you may have heard of is the coccidia. And coccidia are creating, particularly the one called Cystoisospora, is creating a lot of problems in indoor conventional farms. As you can see, these are again a number of positive samples in this piglets, weaners, fatteners, and sows. So you can see in the conventional farms, you see it mainly in the piglets, or almost entirely in the piglets. And it's all of this is this Cystoisospora. And this creates a lot of problems. And about, I think in Denmark, about 80% of all the piglets are treated preventively. This is not the case in organic farms. We do find these coccidia in organic farms, but they seem not to create the same problems that are usually seen within the first week of life. So that's a very positive thing. Even if we have the parasites, it seems that they don't create major problems. So these are the major ones that we are dealing with. Lastly, we also have ectoparasites. And for this, I can only say that under Danish conditions, these are very rarely observed, and they have not been reintroduced to the commercial organic farms. They are not seen in conventional farms, very, very seldom. And apparently they have not been, even if organic farms are more open, we have not spotted them, not yet in organic farms. It might be different in other countries. So the major ones are the blood-sucking lice, and then of course pig mange or swine mange. So I'm not going to say much more about ectoparasites. But if we go to the endoparasites, we have different approaches to the control. One is to sort of act on the environment to reduce the contact between host and infective stages. You can do that by pasture management. I'll get into that. And also hygiene principles. You can manage your litter in the right way, or you can do biological control. Another way that you can look on increasing the host response against the parasites. That could be by vaccines, genetic selection, or it could be by well-balanced nutrition. And then lastly, you can eliminate or modulate the biology of the parasites by manipulating the diet of the pigs. And this is something I will address later on. That could be by phytotherapy, or you could also use commercial antiparasitic drugs in a better way. I will not get into that because this is about alternatives. On pasture and Danish organic farms, which is what I know best, we have farrowing paddock. They then either stay on sort of starter pig pastures, or they go into pens where you have the starter pigs. Later they move on to finisher pig areas, and then ultimately they go to slaughter, of course. And what we can see is that the contamination levels on pasture are very high on these starter pig pastures. This is number of Ascaris eggs that you find on per kilo dry soil that we have examined. It's much, much lower on the farrowing paddocks with the lactating sows and also with the dry sows. But where the starter pigs are, you tend to have very high levels, and this will cause most of the weaners when they go inside, or the starter pigs when they go inside, they will already be heavily infected with Ascaris. The problem of controlling these pasture infections or doing pasture hygiene is that there is a very long survival of Ascaris eggs and also of the whipworm trichuris. This is a major obstacle to pasture management. As you may know, that you can shift every second year in ruminant farming, and then you can get rid of parasites in this way. But this is very difficult in this case. As you can see, these red columns here indicate number of Ascaris that they pick up year after this particular area was contaminated in 2002. They were contaminated for just a month's time, and then they were followed on for, as you can see, up to 13 years after. Still 13 years after contamination, we still find contamination on the pasture, and the pigs will get infected. So it's difficult to design a pasture rotation scheme here for pigs, and that's what a lot of people have experienced. When we get inside, it's typical. This is a typical deep litter system we have here. We found out when we looked into this that we have these latrine areas where they defecate. We have some clean areas where they lie down, and then intermediate areas sort of in between. And what happens is, as you can see here, this is number of eggs that we find in each section. This is not per kilogram of litter, but it's just the total number of eggs. You can see there's an enormous amount of eggs in the latrine area, and then in the clean area, there's also eggs, but much fewer. But the case is that a lot of them, or more of them, are embryonated and they're ready to infect. So actually you have high levels of contamination all over your indoor pen. And this is what these are sort of figures showing the same. So for that reason, it's very, very important that you get when you get rid of your litter, that you somehow sanitize the litter, either by composting. This is what you see here. This is a dung litter heap, traditional dung litter heap, or you put them into a biogas thing. In slurry, the eggs of Ascaris will survive for a long time, almost a year, we have shown clearly. But if they go into biogas, or if they're used, or if they're composted in these deep litter heaps, or like this, I've shown a traditional, there you will see the temperature goes up very high. This is the temperature here, and you can see in the core the temperature reach more than 60 degrees within a week's time, or even faster than that. And by doing that, all the eggs will die, and you will sanitize it. You have to move it around because on the surface they do not reach this temperature, so you have to turn your heap around after a week's time, and then you have all the eggs removed. So either this or or biogas, and then take it out to a pasture or a crop where you do not have grazing pigs. That's very important. So use it on your crops. That will be the way to do it. You can also get, we have done experiments in order to get rid of the eggs. I mean, that would be marvelous if we had some kind of means, for example, biological control, that we could spread something on the pasture and then get rid of the eggs by, in this case, we have looked into some of these fungi that eat eggs. So these ones are simply, these fungi grow, as you can see, in the feces, and they enter the eggs and they kill the eggs. And by doing this, you can get rid of the contamination. So we thought this was an excellent way to do it. And the studies we didn't do with pig parasites, but we actually did it with another roundworm from poultry that looks a little like Ascaris from pigs. It looked very promising when we did the in vitro studies, but as you can see here, when we use sterile soil, or when we took it out to real soil, when we use sterile soil, this is the first one. You can see that there was, in this case, there was without fungi. Then you can, with this column B, or the column here is F, that's where we added the fungi, and the fungi were eating the eggs, and you can see the contamination went down to from more than 400 to less than 100. So this was very nicely, but when we had non-sterile, we were using this or doing this in sterile soil, but when we use normal soil, then there's a lot of competition from other fungi. And in this particular case, this fungi that we were using, this Pucconia we were using, it was not able to compete with the other fungi. Therefore, we had no effect. So you can see here, non-sterile soil with eggs and non-sterile soil with fungi and eggs. There's no killing of the eggs. So basically, in principle, it could work, but right now we do not have the any kind of fungi that we can use in the control of or biological control of these eggs. Breeding for resistance is something that we have not tried to do in practice, but we have looked into the heritability of egg counts and in infections. And these are the heritability of egg counts for Ascaris, for the roundworm, and for Trichuris, the whipworm, a study we did where we infected 200 animals, and we knew the mother and we knew the father. And as you can see, the heritability is something like 0.3. And even at some stages during infection, we had heritabilities of the egg counts of 0.7. So that means that about 70% of the variation can be explained by the contributions from the sow and the boar. So that's a lot, and it's a level where you can easily do selective breeding like you do for any other characteristics. And we actually did find this SNP that is responsible for this, or we found one SNP that was associated with resistance. And we did a study where we had the homozygote for the susceptible genotype, susceptible haplotype, and then we have this heterozygote one, the ABs. Unfortunately, we couldn't make the BBs because we couldn't get it. But as you can see clearly, that we had much lower worm burdens in this homozygote than in the heterozygote, but it was only marginally significant. So what we actually know is that there will be pigs that are resistant and pigs that are more susceptible. And it is feasible to do some kind of selective breeding if you want to do that. But to my knowledge, we haven't done that. Then lastly, the approach we could use is that we can sort of manipulate the diet by using some kind of bioactive crops that are included in the feed. There's a lot of compounds, bioactive compounds, alkaloids, phenolics, for example, tannins, terpenoids. Some of them are used in traditional veterinary medicine, And there's a lot of in vitro studies, but very few in vivo studies. And therefore we need a solid scientific validation before we really recommend this. I'm sorry to say that, but we worked a lot on this, and some of them are very promising. What we've seen is that in the ruminants, we've seen condensed tannins are very promising. They also seem to work in vitro in pigs, but we have not shown it in vivo. We worked with fatty acids from seaweed, so seaweed supplementation. And then we have worked with fermentable carbohydrates. I'll just give you a few examples of this. This study, we used seaweed in combination with rapeseed that was fermented. We were adding this to the diet. We knew beforehand that the in vitro study showed a high antiparasitic activity of seaweed from these fatty acids. That's a traditional omega-3 and 6 fatty acids from marine products. And then we did a study in 4 sub-studies in an outdoor farm. And we used 2 times 25 weaners in triplicates. These studies here, sub-study 2, 3, and 4. And you can see in all cases, when we looked at the overall excretion of eggs, it was lower in the groups getting seaweed. Unfortunately, we did have some negative effects on the weight gain. So we have sort of to adjust the dose of this so that we get the benefits and of course not all the disadvantages. This was Ascaris suum, so this was the roundworm. Against Esophagostum, the nodular worm, we have seen very nice results by adding 10 to 15% inulin from chicory roots that we were grown organically and just feeding to the pigs, infected pigs with having nodular worms. And what you can see here is the fecal egg count. When we added this chicory inulin, we had almost 0 egg counts for the entire period, whereas in the controls where they did not get chicory with inulin, you can see that we have the normal pattern of esophagostomum infection. So I would say that we know by giving these fermentable carbohydrates, we can control infections with esophagostomum. Unfortunately, it does not really work in the same way for Ascaris and Trichuris. Also, the inulin affects the immunity, and it actually has strong interactions with the Trichuris infection. In this case here we did, and that means that we have a high, very strong regulation of the Th2 genes. This also has implications for other infections, but this is something that has to be looked into more. I think this is a very interesting finding also. So lastly, we are also looking at now in a new project called Roam Free. We're looking into combining agroforestry and pig production. And you can see this is where we will, for example, look at salix, willow that will grow in these paddocks and see if, whether they have any consumption or whether they eat this, or we'll also try to put it in the fodder. And we know from in vitro studies that salix has a lot of good potential for antiparasitic activity. And these are studies that we're doing together with Slovenia, Romania, Norway, and Italy. So lastly, the perspectives for this, I think there's a lot of evidence that suggests that we have higher levels of endoparasitic challenges in organic pigs compared to the conventional. It's not known whether this actually translates to greater impact on health and production. But in general, I would say that some means of control is recommended. The efficacy of pasture management by rotation, as we can do as an extremely efficient way of control in ruminants, is really hampered by these long-lived eggs that we see in pigs. And resting for several years is needed, and we simply have problems telling how many years should you rest a paddock or spell a paddock until it's going to be reused. For sure, it will require quite a lot of land for the farmer, but we are talking resting a pasture for 3 to 4 years in order to get any significant reduction in the pasture contamination. Safe removal and sanitation of litter is very important. It should never go out into areas where you're going to have pigs later on. And the change of litter and cleaning of indoor pens between groups is time consuming, but it's also worthwhile because we know there are lots of contamination indoor. Using the bioactive feeds, I would like really to recommend that. And I think it has huge potential for the nematode control, but basically we are not there yet, I would say. Methods of monitoring and targeted interventions are something which is also buzzwords in the ruminant sector. And I would really like to be able to say how to monitor and then how to target the interventions. These interventions would probably have to be with drugs, and that is sort of not really what we want in this context. But of course, compared to treating the whole flock, it might be a good idea perhaps that that you restrict to certain groups. There's still a big problem, of course, is the heavy environmental contamination that we do see in all these pig areas, and therefore it is much more difficult to do sort of targeted or selective interventions in pig farming. So this is something we have to work on if we are going down this lane. And then lastly, some other perspectives on parasitism. I mean, there are studies of people who claim that, that parasites, you can look at parasites as a natural part of any ecosystems, and each species have this potentially useful information unit, and a healthy ecosystem have healthy parasites. And biodynamic farmers, as far as I know, consider intestinal parasites as earthworms of the gut. This is something, of course, that we are also very much aware of, and we know that lack of exposure to helminths may be related to autoimmunity, as this is what we call the hygiene hypothesis. And this has been suggested in humans, that, as you can see, a lot of these immune-related or autoimmune-related diseases are really in industrial societies increasing dramatically. And the hygiene hypothesis is that we are simply living in environments that are too clean. And one of the things that we actually miss in the modern society is helminths, actually. A balanced, diverse nematode population is contributing to a normal gut microbiota and might thus play a role in this immunological homeostasis. This is sort of one of the paradigm. I think there's a lot of truth in this, and I think that, but at the same time, I mean, we should not disregard all the problems that the parasites cause. So finding this right balance is in organic pig production is really one of the future challenges. So I would like to thank a lot of projects that have contributed this over the years, and also a lot of PhD students who have done a lot of this work, and we're very grateful for this. So thank you very much for your attention. Thank you.
Hello, everybody. My name is Spiridoula Athanasiadou, and I am a research scientist from SRUC in Scotland. And I will be presenting you on some work we have recently completed in a Horizon 2020 project on the replacement of anthelmintics in European organic livestock. So the presentation today, I'm going to start with giving you a little bit of a background on helminth infections in ruminants. Then I will go to report on some work we did in RELAX on current use of anthelmintic drug in Europe. I will introduce you to the alternatives we have been investigating, the bioactive plants and the nematophagous fungi, and then discuss the on-farm implementation and give you some first glance, if you want, on some recently completed on-farm validation trials. Parasitic gastroenteritis is one of the biggest health challenges for small ruminants. For that purpose, gastrointestinal nematodes are of most economic importance. mostly because they cause subclinical disease. The most important species that are relevant for subclinical disease, particularly in Europe, are the abomasal species Teladorsagia circumcincta and Amoncus contortus, whereas from intestinal species it's Trichostrongylus colubriformis and Nematodyrus batus. In terms of some subclinical disease, the penalties they are resulting are in weight gain, milk production in milk-producing animals, of course, wool growth, and carcass quality. And there are data to suggest that losses are affecting around 50% of farms globally, and the prevalence of these parasites is almost to 100% in small ruminant farms around the globe. So in small ruminants and in large ruminants, actually, the gastrointestinal parasite control is traditionally achieved with anthelmintic drugs. And I have included this figure here, which I really like to discuss. I mean, it is a figure, it's 40-year-old now, but it's still very relevant, and it shows how expensive gastrointestinal nematode challenge is. So if you have a look at this figure where the y-axis shows the lamb weight gain and the x-axis shows weeks, and you see the blue line at the top, this is the growth curve of an animal that has absolutely no parasite challenge. And then you see the bottom line with the open circles, where these animals are growing when they're infected with about 5,000 larvae a day. And this is the kind of usual, if you want, or standard average parasite intake for animals that are grazing. So you see what a big loss it is from the no challenge to 5,000 larvae a day. And then you see the red line, and the red line shows that these are the same animals that take about 5,000 larvae parasites a day, but they're drenched with an anthelmintic drug. And they're drenched every 3 weeks. So what this figure shows is that when you have the red line, so you're drenching the animals, that regular drenching will improve lamb growth. So your animals will grow better if you drench them when they're parasitized. However, if you look the difference between the red line and the blue line, it is clear that drenched and challenged animals will still lose out. They still grow much less compared to the blue. Now, if you check the second line from the top, where the animals are having about 1,000 larvae a day, then you see that these animals are losing out much less compared to those that are taking 5,000 larvae a day and they're drenched. So maybe the key is to reducing the impact of parasitism is just to reduce the challenge, and then the productivity is going to be improved. So yes, anthelmintic drugs will work, however, not to the degree that would be required. And in addition, we have the organic and the low input farming, which is requiring sustainability without the excessive use and the regular drenching with anthelmintics. And of course, there is this rise in anthelmintic resistance, which means that the drugs don't really work as well as they were supposed to. And all these factors are telling us that we need to find other ways of managing control problems, and then yet reduce the use of anthelmintics for parasite control. So in relation to the organic farming, the first thing we wanted to do in RELAX project was to see how much anthelmintics are actually being used, how much drenching is taking place in organic farms. So we designed a survey, and the survey was addressed to organic experts of all livestock species, and it was translated and disseminated by RELAX partners throughout the EU and outside EU, across Europe. And we had 2 questions that we were asking people to answer. The number of farmers that request anthelmintics in their health plans. So this is the expected gastrointestinal nematode risk, because all organic farms are requested to do a health plan for their animals, and in their anthelmintic use is one of the drugs they're asking permission for. And the second is the number of farmers that require a supplementary request for the use of anthelmintics. So this is when gastrointestinal nematodes risk is greater than expected. So in addition to scheduled, if you want, use of anthelmintics, how much more extra are people asking to use? And these are the two questions we ask organic experts to answer. And we got 139 responses, which is covering 16 countries in Europe. And the people who responded were responsible for more than 17,500 farms around Europe. So this is the percentage of organic farmers that are asking for anthelmintics to be included in their health plan. Remember, these are the organic advisors that are giving this data. And there is a large variation in the planned use of anthelmintics around Europe, with 100% of organic farmers asking for the use of anthelmintics in their health plan in Greece, which is the right-hand side of the graph, and a very, very small percentage of organic farmers in Lithuania that are asking for anthelmintics to be included in their health plan. So that shows that there is a lot of variation in the expected risk of gastrointestinal nematodes in European countries. And here, in addition to the percentage of organic farmers that require anthelmintics to be included in their health plan, that's the blue bars, we also have the percentage of organic farmers that require supplementary use of anthelmintics. And again, you see that there is a lot of variation. I suppose not surprisingly, you see in Greece, for example, where 100% of the farmers asked for anthelmintics to go in their plan, there's no requirement, there's no need for additional supplementary requests. Whereas in other countries like Romania, for example, there is a large percentage of farmers that require anthelmintics in their health plan, but also a large proportion of farmers that require additional use of anthelmintics. Again, a lot of variation and a lot of different responses across the 16 European countries. So what this survey showed us is the big variation in the use of anthelmintics across Europe, but also that it is hard to predict disease because in some cases, although there was a lot of anthelmintics used in the health plan, there were still requirements for a lot of supplementary requests. So that requires ongoing flexibility for the use of anthelmintics in organic systems in order to maintain the health and welfare of organic livestock. Some countries appear to have more effective prevention of parasites, or they have a lower risk of parasites. And for sure, there is variation in the gastrointestinal nematode threat, which depends on geography and climate and data limitations, of course, etc. So we have calculated, based on this data, we have made some estimates that for each organic livestock per year, 0.86 anthelmintic treatments goes in the system. And if we multiply that with the total heads of livestock in EU, that gives around 50, which is around 53 million. That approximates to about 45.5 million anthelmintic treatments per year that goes in the EU. That gives an estimate of how big the problem is, if you want, or if it is a problem in any case. So how can we reduce the requirement for anthelmintics drugs? And this is where the alternatives come in. So there are a number of grazing strategies that are being developed. The use of optimized nutrition at times where animals require extra nutrients. Breeding for resistance is a method that is being developed. The use of bioactive plants for parasite control, and of course the biological control. So before we go into the alternatives, I thought I will present you here with the gastrointestinal nematode life cycle. So we have the sheep with the adult worms in their gastrointestinal tract, and they are producing the eggs, which are excreted in the environment. And the eggs are developing into first stage and second stage and third stage larvae, which are the infective larvae, and these are consumed again by the sheep to complete the life cycle. So we understand that there are two kind of environments in the life cycle of gastrointestinal nematodes: the parasitic stage, where the parasites are inside the animal, and then we will need to look for alternatives to reduce the gastrointestinal nematodes in the animal, but also the free living, which is the infective stages that are outside the animal. And then we have to find ways, or we can find ways to reduce gastrointestinal nematode on pasture. And the RELAX project looked at both of these stages, about an alternative that can reduce gastrointestinal nematodes in the animal, but also in the environment. So the first alternative that RELAX has been looking at, and I wanted to present you, is the use of bioactive plants. So when we talk about bioactive plants, these are plants that their intake can result in antiparasitic activity. And I have some examples of bioactive plants here. Chicory is in the first picture, and we at SRUC have done a lot of work on chicory. Sainfoin in the middle picture, and it's mostly our colleagues in France, in INRA, they have done a lot of work with sainfoin. And lotus is a plant that has been researched extensively in New Zealand and Australia. I chose to present a few of the benefits of grazing on chicory because, as I said, this is a lot of work we are responsible for, and we've done a lot of experiments here in Scotland. And grazing on chicory has multiple benefits. It results in fewer worms and faster growth in lambs. Also reduced drug input and as a consequence, of course, a reduced parasite challenge. So you see here on the image on the left where we've got the lamb gain on the y-axis, whereas on the x-axis is ewe treatment at turnout. This is a study from with ewes. And you see that in the absence of an anthelmintic, which is the first two bars, no anthelmintic given there, animals on chicory are growing much better compared to those that are grazing on grass clover. On the other hand side, on the top right corner, you see the fecal egg count graph, where animals on grass clover are having higher fecal egg counts. So these are the number of eggs in the feces of animals, parasite eggs, whereas animals that are grazing on chicory, the black line, have much lower fecal egg counts compared to the control. And then the final figure at the bottom, it shows the drench numbers per animal. And you see that the animals on chicory have lower requirement for drug compared to those on grass clover control pasture. So a lot of benefits for grazing on chicory. In RELAX, we chose to research a different bioactive forage, and this is heather. How does it work against gastrointestinal nematodes? So we're not sure yet. We know that heather contains condensed tannins, and we also know that past research has demonstrated that condensed tannins have a significant anthelmintic effect. We've also seen previously that goats that were grazing on heather reduced their parasite burden. And we also know that hill sheep are able to graze on heather leaves. So we've seen that. This is something you see if you just drive in the Scottish countryside. I mentioned that heather has condensed tannins, and condensed tannins are plant secondary metabolites. And you see the chemical formula of condensed tannins here. But what is important is that condensed tannins have the potential to have direct or indirect effects against parasites. So when we're talking about direct effects, this includes changes in the parasite development or parasite survival. And there has been studies that have shown that condensed tannins can have an impact on the exsheathment of L3s. So L3s, which is the infective larvae of the parasites, need to go through an exsheathment phase in order to be able to become to establish in the animal, and condensed tannins are having an impact on that. Also, they have the ability to stop egg hatching and to stop larval development and to kill adult worms. But in addition to these direct effects, direct killing of the parasites, condensed tannins also have the potential to have an indirect effect by increasing the intestinal protein supply, which effectively benefits the immune system of the animal. And that has an impact on plasma antibodies and mucosal mast cells and globular leukocytes, which are white blood cells which are effectively active against parasites. So condensed tannins have the potential to do direct kill worms, but also to improve the immune system of the host and help the host deal better with the parasites. So the first thing we did in RELAX was to do some in vitro testing of heather to show whether heather extracts can have an impact against parasites in the lab. And we tested 2 different stages of parasite, the eggs and the L3s, the egg hatching and the L3 motility. And we used different types of extracts. We used extracts that came from 5 different countries, Scotland, Switzerland, Norway, Germany, and Spain, from 2 different seasons, spring and winter. Also, we tested their efficacy against 2 different parasite nematodes, one abomasal and one intestinal. And also, we had the ability to test different species of heather and different doses of heather. And we observed that there was variation in the efficacy of heather extracts depending on the country. So some countries had higher efficacy. For example, heather from the UK had higher efficacy compared to heather from Norway. But we also observed that the different parasite species have different susceptibility to heather, with Teledorsadia circumscripta showing to be more susceptible compared to the intestinal species. And a seasonal effect was evident. Overall, here this is a table of the LD50s of heather extracts when they were tested against Trigostrongylus colubriformis, that's the intestinal nematode. And the lower the LD50, the stronger the extract. So you see quite consistently that the heather that was collected in springtime had higher efficacy compared to that from the winter time. Certain areas, for example, spring in Germany was significantly more efficacious against T. colubriformis compared to winter, the winter extracts from Germany. So a lot of variation, both in terms of seasonality and interactions with the country. So the conclusions from our in vitro testing was heather extracts varied in their anthelmintic efficacy, but also the nematode species showed variable susceptibility to heather extracts. The next steps was to associate the efficacy of the extract with the structure or the content of condensed tannins, validate the in vitro findings with in vivo, and also investigate possible detrimental effects of heather on performance. So the second alternative that we investigated in RELAX was the use of nematophagous fungi. This is an image of the fungus attacking one of the worms, and the fungus is called Addingtonia flagrans. And we tested fungi efficacy both in vitro and in vivo. So the fungi, it works on the environment. It's attacking the larvae when they're outside the animal, so in the fecal pellet, and it is fed to the animal as a supplement together with some food. The fungi can kill the free-living stages of the parasite, and previous studies have shown that it can reduce pasture contamination by about 70%. They are commercially available in some countries, for example in Australia. And what we tested there was the interaction of the fungi with the heather diet to optimize efficacy. And we tested fungi on farm as well to see how well they would work on farm. And we showed that when animals were supplemented with the fungi, and you see what the results are here, there was a reduction in larval recovery compared to the control animals when they were not fed fungi. So if you see on the y-axis here is larval recovery, the percentage, and on the x-axis were the 4 different groups of animals that were either fed no fungi and fungi in a control diet, and no fungi and fungi in a heather diet, because we're also interested to see if there was any interactions between heather and fungi in terms of having a possible inhibitory effect from heather on the fungi or anything like that. We didn't see any interactive effects, but we did see that when the fungi were fed on the animals as expected, as it has been shown before, there was a significant reduction in larval recovery when the animals were eating fungi compared to control animals. And we also looked at how heather resulted in any changes in the egg output of animals that were eating it. And what we show here is that as the time progressed and animals continued eating heather, they had a reduction in their fecal egg output. On the y-axis here is the logarithmic egg output, and on the x-axis is the date. So in this trial, the animals were ingesting heather for a period of almost, I think, 3 or 4 weeks. And it seemed to be an effect that was maintained for the whole period that the animals were grazing heather. They showed a reduction in their egg output. And after we had seen that heather worked in vitro in the lab, but also in control conditions in SRUC's experimental farm and also other partners' experimental farms, we wanted to see what happens on real conditions, so on real farms. And we recruited organic farmers in 3 different countries in Europe, and we asked them to split their animals in 2. One is the treatment group, where animals would be receiving either the fungi or the heather, and the other one is the control group, where animals would be managed according to their farmer standard practice. We would ask that in the treatment group, the animals that are grazing heather, that would have access to heather every day. And for the animals that were taking the fungi supplementation, that they would take that at a rate of 1 gram of spores per 100 kilogram body weight, and this would be added in their feed daily. And then we took a number of measurements just immediately before the trial started and at the end of the trial period, which lasted between 6 to 8 weeks, depending on the farm. So we would do an initial and end point fecal egg count. We would ask for body weight and or body condition score. And would also take health information, including any anthelmintic treatments that were applied. And very importantly, we wanted to see, to get the farmers' experience of the alternative and what was their view, possible constraints, logistic problems, etc. So our results showed that there was no major impact of either heather or fungi administration on the fecal egg counts or performance of the animals. That's no major impact negative and no major impact positive. Overall, there was a small number of sheep that was managed in these farms. So it was the people that came forward to volunteer for these trials had a small number of animals available, so it was easy for them to manage. We got a significant feedback on the use of alternatives and possible constraints and also gains. And we are going to be using this feedback to streamline the use of alternatives. We had people that were very happy to take part and other people that recognized that it took a longer management process in order to incorporate either the heather or the fungi on their planning. So the conclusions on the farm trials were that the experience was variable, as I mentioned before. Almost everybody recognized that there was an increase in management time that may be required. There was no significant additional costs, although it must be said that some of the costs for the experiments were met by the project. Despite the fact that some farmers recognized that it would take longer maybe to manage their animals, they were still interested to try and reduce their anthelmintic input. Which, however, was already quite low in the farms that volunteered to take part in the trial. So clearly these are people who are very interested in keeping their anthelmintic input low, and that's why they're interested in alternatives. But for them in particular, it didn't appear to be a significant need to reduce the anthelmintic input because it was already quite low. So the overall conclusions that I wanted to make and close this presentation is that bioactive plants can certainly contribute to reduced use of anthelmintics. Overall, alternatives to anthelmintics, including bioactive plants and nematophagous fungi, can be used for parasite control. However, they should be seen as complementary to anthelmintics rather than instead of completely replacing anthelmintics. And the way that's probably should be seen by farmers, but also by advisors, is that this is just some options from a basket of options which are available to the farmer to use in order to improve implementation of the alternatives. So seeing it as a tool that is available and can be used if suitable to your conditions. So thank you to all for your attention, and I wanted to thank our collaborators in RELAX, but also our funders, the European Union and also the Scottish Government, in particular for SRUC funding, and I'm available for any questions you may have. Thank you.
Hello, everyone. My name is Lucius Tamm. I'm coordinator of RELAX, of the RELAX project, and I'm happy to tell you a few things of what we've achieved and can contribute to our overall objectives. RELAX stands for Replacement of Contentious Inputs in Organic Farming Systems. It's a sister project to Organic Plus. Our objective was to evaluate solutions and to further reduce the use of external inputs and to develop and adopt cost-efficient and environmentally safe tools and technologies too, and that's the important point, to reduce the use of copper and mineral oil in plant protection, to identify sustainable sources of plant nutrition, and to provide solutions to support livestock health and welfare. And our concept from the beginning was, of course, to build on results of previous research projects and to take far advanced solutions forward. So that's the underlying concept. We have included 13 European countries of academia, research institutions, but more importantly also many farmers associations to do on-farm research. So we have quite a large-scale consortium involving many stakeholders from different groups. Okay, this is the group photo of our consortium when we started off, and all the contributing partners there, including one of Spain. But I would like to put our activities in a broader context related to what we need to do to move forward with organic farming. So as you all know, there are some global challenges for agriculture. So like soil degradation and climate change, water use and pollution, but also loss of biodiversity. But nowadays also in the political situation, the yield security issue has become very important again. So we have to comply and contribute to these overall objectives to provide yields, but also to reduce the environmental impact. And as you all know, organic farming contributes or performs quite well if it comes to productivity in subsistence and smallholder farms. It creates added value in food chains. It has all sorts of benefits to the environment. But there is the famous yield gap. And this is one of the problems we are facing and which we have to address. And I'll come to that in a second. The benefits of organic farming and its potential contribution to agriculture have been recognized at the EU level and in many member states, in particular in Europe. And therefore, the Farm to Fork strategy foresees very ambitious goals to upscale organic farming to 25% until 2030. And what you can see here in this graph, this means a tremendous increase of the surface in a short period of time. And this puts all sorts of challenges to us, to the actors on how to achieve this transition and how to achieve these objectives. It's logistics, it's training, it's market all aspects of the organic sector are involved. So when we talk about challenges to organic farming, then I would say we need to increase productivity to close the yield gap while retaining the benefits to the environment. So we have to avoid the collateral damage to the environment. We also have to replace problematic practices, and this is what Organic Plus and Relax are about. And we also have to find ways to allow for upscaling for organic farming. So that's also a major challenge in logistics. The timeline of Relax was when we started off. We started off with data collection. You know, where do we stand? What's the state of the art? We identified the needs in surveys. What is used right now? What are the alternatives? We did develop and customize available alternatives. We tested them on farm in various countries and contexts. We also looked at the socio-economic, environmental, and acceptance issues for novel solutions. And we've also developed roadmaps to be presented to the European Union on how to implement those alternatives if it's possible, and on how to reduce the use of contentious inputs. The state of play is, yes, we see, in a nutshell, yes, we see that there are alternative tools and technologies, and they may contribute to the reduction of these contentious inputs in many cases, not all. The transition to improved management practices has to involve all players along the value chain. That's totally clear. There are very different, there is a different potential for introduction of alternatives, but there are also different bottlenecks in each case which may hinder the transition. So they need to be identified in each case. We need tailored support strategies from the policy side, because else nothing will change. We also identified actions that are needed at the EU level to encourage the use of alternatives and to encourage the reduction of contentious inputs. And as I said, we have drafted some roadmaps to reduce the use of contentious inputs. And those roadmaps, they have to be fair, reliable, and also implementable. It's no use to claim everything is fine if there are serious problems. What I want to raise here is we've addressed some aspects, but if we want to improve organic farming and introduce further innovations in organic farming, really to address the demands from the world, to feed the world and so on, then it's not only about technological innovation. It's really also about social innovation and ecological innovation. And these are aspects we have not addressed in Relax because that was not the purpose. But we have to keep in mind that these other avenues or roads for innovation need to be addressed too in order to achieve major progress for organic farming and agriculture in general. I'm going to give you some of the key results and key recommendations coming from Relax related to anthelmintics, antibiotics, and synthetic vitamins. It's really quite condensed. In order to replace or reduce the use of contentious anthelmintics, we were following two approaches. One was the use of a biocontrol agent, Didingtonia flagrans, which actually reduces the pests in the dung and not in the animal. So it's fed to the animals, but it's really working once they have excreted then in the field. So it's so to speak, to decontaminate the pastures. The other approach is to feed them reducing feedstuffs such as heather. So what we found is that both heather and Dudingtonia flagrans have shown a lot of potential to reduce the use of anthelmintics, but there are not veterinary drugs that will completely replace anthelmintics. It's rather a complementary strategy to reduce the overall pressure on pastures. What we also see and realize is the registration process for herbal products such as heather as feed additives, but also of any veterinary medicinal is very lengthy and cost intensive. What we've also seen as a sort of an obstacle or something we have to think about is that veterinarians need to be included, those people who look after the health of the livestock, in the whole process, because it's important that they accept the technology and that they claim some ownership on it, and so that they can also combine it with the use of anthelmintics as a curative, but the use of the preventive strategies as the protective strategy. What's also quite clear is that to use those technologies needs quite a bit of know-how. So it's necessary to provide both technical but also financial support for the farmers. Otherwise, the adoption of these strategies will take a lot more time, or will probably be not be possible at all. As regards antibiotics use in dairy production, so we only used on dairy cattle, cows. We have also followed two approaches and explored two approaches. One is this AHWP, which Animal Health and Welfare Protocol. That's a way to look at the animal's health in a holistic way, including the feeding regime, including all sanitary methods, hygiene, and so forth. So the other approach was the use of essential oils. So the Animal Health and Welfare Protocol, which is really developed and used together with the farmers, it's very important. It's to work very closely together, veterinarians and the farmers. It shows a very good potential to reduce the use of antibiotics in mastitis treatments in the short term. What it needs, it's knowledge intensive, so it needs sufficient investment in advisory structures. That's pretty clear, but it shows really good results and the drastic reduction of the dependency on the antibiotics. On the long term, medium and long term, we might think that these reduction strategies could be complemented by the use of essential oils. But here we see that we haven't finished this yet. What we see is that we have no negative impact of essential oil use on the milk quality parameters. We see no difference between standard antibiotics treated cows or essential oil treated cows. That's quite interesting, but there is still a lot of work to be done. So we need to continue some of the research. Again, the registration process for these products is extremely tedious and difficult. And the legal status of essential oils and other natural treatments is so in some places more problematic than in others. Again, this is absolutely crucial if we think about the health management of the livestock. The veterinarians need to be included from the beginning when introducing such strategies in order to increase acceptance. And especially in the case of the Animal Health and Welfare Protocol, the veterinarians are just crucial to have them on board because of their knowledge, because those are also persons to drive it. As regards the use of the synthetic or GMO vitamins, we followed two approaches. One was we questioned whether the dietary prescriptions or recommendations are really appropriate. So we've tried to, we've established that the input or the feed, the amount needed to keep the animals healthy it can be much reduced. So it's not necessary to supply the full dose, which is currently recommended in standard recommendations. So there is a high potential to reduce the use of vitamin E and vitamin B2 to start with. We also see there is no way to phase vitamins out completely without, I mean, the addition to feed to reduce it completely, because it would endanger the animal health. So that's not an option, because it's not good for the animal. But we see some reduction potential. So for vitamin E in dairy cows, we believe about 50% could be less could be used, and in organic poultry, we believe about 30 to 50% less could be supplied in the feedstuff. These reductions, based on our current knowledge, they could take place in the very short term. There is no technical constraints for the feed industry that can just do it if they have the demand for it. If we want to reduce the synthetic vitamins or GM vitamins further, then again, there is an option to study the level of native vitamins in some raw materials of organic animal diets. It's unknown, I'm told, at this stage. We've also seen that supply from controllable European sources of vitamin B2, which is clearly not produced with the help of GMOs, is possible and it's existing. At this time, it's a bit delicate from the political point of view, because there is only one company as a supplier. So in order to increase the acceptance for this measure, it would be quite beneficial to have more than one supplier in place. The alternative is also a little bit more costly, but it doesn't affect the production costs, the overall production costs at all. So we believe we should further develop the market and stimulate competition among the companies, and this also in order to avoid any shortage in vitamin B2. Just because one company stops manufacturing this stuff, then the industry has a major problem. And another recommendation is also the European Commission has to take a very clear decision on the regulatory status of products consisting of vitamin B2. So they should be without the help of GMO. They should be quite clearly preferred. And they should also facilitate the registration of these alternatives at the EU level, which is, again, registration is a bottleneck. And there is an open question whether alternatives should be registered as feedstuff or feed additives. This has quite some implications at the registration level. You can find a lot of upcoming information on our website as they come and as they become available. We are now at the end of the project, and the information comes out. You will also find quite a lot of scientific publications there coming out of the project, and we will have detailed report on the alternatives and the potential. There are detailed reports on the reduction strategies and reduction options, and there are also some practice abstracts that are already online or which become available quite soon. So a practice abstract from our project looks a bit like this. It's the example of a copper replacement. So it's an easily digestible 2-page information leaflet. It's there on vitamins. I've seen the proof print of the anthelmintics, and the other ones will be soon available. The detailed reports on the roadmaps for reduction of contentious inputs are quite detailed reports. It's up to 30 pages or so, and you will have them get them. We will make them available as soon as they are authorized by the Commission. So thank you for your attention, and please feel free to contact us or my colleagues after the workshop via email or whatever channels you want to choose. Thank you.
Hola a tots. Moltes gràcies a tots els ponents. Ha arribat el moment de les qüestions. Ara podeu escriure les preguntes en el xat que us apareixen a la pantalla, a la part de baix. I, sisplau, si podeu indicar a qui va dirigida la pregunta, ens facilita molt després a l'hora de repartir-ho i a la resposta. Hi, now it's your turn to ask. You can type the questions in the chat that will appear in the bottom side of the screen. And please indicate to whom the question is addressed in order to facilitate the answer. Avui tenim amb nosaltres connectats la Spiridoula, la Berit i l'Stig. Hi, everybody. Today we have connected with us Berit Blomstrand, Spiridoula Athanasiadou i Stig Thamsborg. I després també la Marta Bojar, que com els altres dies ens farà les traduccions. And Marta Bojar as well. Hi, Marta. That, like the other days, she will do the translation. I think that's a little bit all. Okay. So, well, sometimes it takes a little bit of time people to write down the questions. Hi, Spiridoula. How are you? I think you should connect your microphone. It's not connected. El micròfon de l'Spiridoula no està connectat i li indico una mica com fer-ho. Perfect. Yeah. Yeah, that's lovely. Doncs esperarem una mica si us animeu a fer alguna pregunta sobre les participacions per no estar aquí tots callats. Una de les coses que a mi em va sorprendre, volia comentar-li a l'Stig, em va sorprendre però em va semblar molt interessant, va ser que va parlar d'un mínim de paràsits en els estómacs, com una proposta que és una cosa que ens hauríem d'anar acostumant. Jo crec que en el sector ecològic també ens ha passat, ens hem hagut d'anar acostumant a un mínim de plagues per poder mantenir l'equilibri. Stig, I was saying to everybody that one of the things that most surprised me when I was listening reading your presentation and listening to them, that you talk about that it should be a minimum of parasites in the animals to keep this, this level, the equilibrium, I think. Yeah. I don't know if Spiridoula and Berit, they share the same opinion.
Well, Can you hear me? Can you hear me? Can you hear me?
Yeah, we can hear you perfectly. Yes.
Okay. Yes. No, it was just sort of to provoke some of you. I wrote that, but I mean, basically, low levels of parasitism will probably have relatively low impact, and I can only add that in humans, you have been discussing this issue of absence of parasites as a problem because you don't really get triggered the right kind of, they play a role in the immune homeostasis in the gut. And that could be a problem if you have absence of, for example, helminths. So it's all about this old hygiene hypothesis that came up in 89, I think it was. So it's about 30 years ago. So there's all, but that doesn't imply that it's a good thing to have lots of worms. And it doesn't imply that you can actually apply worms to get rid of some of these autoimmune diseases. But I can only say that people have done serious clinical studies to look into this. And so far, some of them have been successful, but in the last 5 years, there's been several large clinical studies in humans, and they have not been successful, so to speak. So that's very brief.
Comenta l'Stig que sí que s'ha demostrat a través d'estudis que es necessita tenir un mínim de paràsits als intestins per tal de mantenir un bon funcionament del sistema immunitari en aquesta zona, que és una cosa que es va estudiant des de fa 30 anys o més.
Thank you, Stig. I don't know, Spiridoula and Berit, if you have any opinion about this subject, or you share Stig's opinion?
Well, I agree. It's better if we have a few worms than none. It's better to have some worms. I agree, for many different reasons. You get the immune system stimulated, you get the homeostasis that Stig was talking about. There are benefits, but of course you can't have many. It's problematic. So we just need to get that balance right. I agree.
Està d'acord, s'ha de tenir una mínima quantitat de cucs perquè això és beneficiós per tots els motius que ha comentat l'Stig, però sí que s'ha de trobar un equilibri perquè tenir-ne massa no és bo.
Yeah, just to comment, I totally agree with Stig. As a veterinarian, I can't really comment on the human health situation. When it comes to livestock, I think it's important to remember or to know or to acknowledge that the animals have to live with some parasites. It should not be our goal to rid the animals of 100% of all the parasites. I don't think it's possible, and I don't think it's wise. So to live with them, I think that's important to acknowledge.
Comenta que ella és veterinària, no sap amb els humans com funciona exactament, però sí que en el tema del ramat és important que els animals convisquin amb els paràsits, que no és bo eliminar al 100% els paràsits i, de fet, no creu que sigui possible. Perdó, algú té el micròfon encès o els altaveus i costa moltíssim sentir bé. Someone has the speaker on, and I hear my voice all the time when I translate, and it's kind of hard.
Should we switch it off when we're not speaking?
I don't know who it is, but yeah, I hear my echo all the time. Yeah, maybe that would be, yeah. Okay, good. Thank you.
Vale, bueno, no hi ha masses preguntes. Aprofito per comentar-vos. Bueno, hi ha una persona que diu que té conills ecològics i que pren nota de tot el dit. There is no much questions. There is some people that, well, a person that says that she has organic rabbits and that she will take note of all the information that you tell today. I només dir-vos que, sobretot amb la part que ha explicat la Berit, el vídeo que vam fer de la visita, o que hem fet de la visita que vàrem fer el passat divendres 17 de juny a l'explotació de la Raquel Servitje, crec que complementa molt amb el que han dit ells i sobretot amb la part que ha explicat la Berit, si esteu interessats en veure casos reals, en ramats reals. Us aconsello, és un vídeo de 30 minuts, tindreu disponible, us l'enviarem tota la informació al RuralCat quan el tinguem i són 30 minuts que són en català i estaran subtitulats a l'anglès. I was just saying that the last week we went to visit a farm here in Catalonia with Raquel Servitje, that is a veterinarian, and we did a video of the visit that did last like 30-35 minutes. It's in Catalan, but we put subtitles in English to make it available to everybody, and that I think that complements quite the exposition that all of you have done, mostly Berit, no? Complements a lot. That's like a real case of all the theory that we have been listening today and that we will send to all of you the links when we will have it available in the web. That I think it should be in a week, 10 days, maybe. Well, no sé, no hi ha més preguntes. Realment, si algú ha escrit una pregunta al principi, se'ns havia quedat al xat de l'última sessió i les hem borrat totes. Si algú havia fet una pregunta superràpida al principi, que la torni a teclejar, sisplau, perquè no s'ha quedat. When we opened the chat, we saw the questions that we had from the other day, so we delayed them all. And maybe if somebody had made a question just at the beginning, we delayed it. So if you can type it again. I si no, doncs, bueno, realment estem complint bastant l'horari perquè a les 11:20 hi havia l'apartat de tancament de tot el curs. Per tant, si en un minut o així no hi ha més preguntes, donarem la sessió per tancada. Well, anyway, the time it's quite the one we put in the program because they say thank you. Because it was 11:20, the moment that Glòria Colom will close the whole course, the cycle of the 9 activities, the 7 talks, sessions, online sessions, and the 2 visits what we did during the whole month of June. I per tant, si no teniu res més a afegir, anirem tancant. La Glòria Colom, que és la persona responsable de tot el cicle de jornades i del projecte Orgànic Plus a l'Escola Agrària de Manresa, ens faria el tancament de les jornades. Well, there is no more questions. If you want to add something or anything more clear after listening your presentations or your colleagues' presentations, now's the moment.
Well, thank you very much for the initiative.
Okay, el Stig diu que gràcies. I don't know, Spiridoula and Berit, if you want to add anything before we close the chat.
No. It was a great initiative, and thank you very much for the opportunity.
Yeah,
thank you.
Thanks a lot. I would like to add one thing which is quite important to me, and that is before treating the animals for parasites, I would like to emphasize the importance of taking fecal samples to check if it's necessary to treat the animals. And this is an important issue in Norway, because in Norway we are really not good enough at taking samples, and we treat without any criticism onto that. So, thank you.
Comenta la Berit que sí que li agradaria afegir que considera que és important que abans de tractar els paràsits s'haurien d'agafar mostres dels fems i analitzar-les. I això, per exemple, a Noruega no s'està fent i creu que és important fer-ho.
Sí, la veritat és que és un comentari molt pertinent. Potser no ha sortit perquè són les típiques coses que donem per suposat, però, òbviament, primer veure si cal tractar i després fer els tractaments. Thank you very much. It was a nice comment because sometimes, Berit, we don't think about these things. We make it like it's what everybody should do, and later on the reality is that we go just straight to the solution without looking which is the real problem. D'acord, doncs despedim a l'Spiridoula, la Berit i l'Stig i a la Marta. Gràcies a tots. Thank you very much all for joining us at this questions and comments slot. I donem la benvinguda a la Glòria, que farà el tancament de la jornada. Thank you.
Thank you. Bye.
Bye-bye.
Bé, ha sigut molt interessant la jornada, gràcies a les ponents. Aquest cicle de jornades ha estat una de les contribucions de l'Escola Agrària de Manresa al projecte Orgànic Plus. Ja durant els 4 anys previs de projecte hem anat organitzant jornades sobre alguns d'aquests temes de les jornades. I una altra contribució important que l'Escola Agrària de Manresa ha fet al projecte Orgànic Plus ha estat una experimentació sobre alternatives a la torba en producció de planter ecològic en col·laboració amb l'IRTA. This lecture cycle has been one of the contributions of Escola Agrària de Manresa to the Organic Plus project. Already, during the previous 4 years of the project, we have been organizing conferences on some of these topics. The other important contribution has been an experimentation on alternatives to peat in seedling production in collaboration with IRTA. Ha estat un honor formar part en aquest gran projecte europeu de recerca i ho hem de valorar i ho valorem com una contribució a la celebració dels 50 anys que fem a la nostra escola enguany. It has been an honor to be a part of this great European research project, Organic Plus, and we value it as a contribution to the celebration of our 50th anniversary of our school that we are celebrating. Finalment, vull agrair la feina dels nostres col·laboradors, la Xènia Torres, la Marta Bujar, que heu vist durant les jornades, i el Xavi López, que no l'heu vist però ha estat fent feina sense ser vist, la feina de la meva companya Joana Amador, la bona predisposició dels i les ponents, tant del nostre projecte com del projecte Relax, i a totes les persones que heu mostrat interès en la millora de la producció ecològica. Finally, I would like to thank the work of our collaborators, Xènia Torres, Marta Bujar, that you have seen in the speaks, and also Xavi López that has been in video production. The work of my colleague Joana Amador, and the good disposition of the speakers of both projects, Organic Plus and Relax project. And to all the people who have shown interest in improving organic production. Thank you and goodbye.